Hi, I'm Mindy, and my project focuses on two genes, foxA2 and spondin1b, and the role that they play in zebrafish spinal cord development. So on the left here, you see a picture of a zebrafish embryo, and this is what they look like about 20 to 24 hours after they're born. And so zebrafish embryos are actually transparent when they're born, which means we can easily visualize the structures within the fish, and that's why I use the zebrafish model. And so during this time, there's a very critical process called neurulation. And that is basically the formation of a neural tube, which is the little blue structure in the diagram to the right. And this structure will eventually give rise to the spinal cord and the brain. So in the neural tube, there's a process called dorsal-ventral spinal cord patterning that's happening. And so a structure at the very bottom called the floor plate, labeled FP on the diagram, this is a primary area of interest for my research. And the floor plate, what it does is, along with the notochord, it secretes a protein called sonic hedgehog. And this sonic hedgehog protein, as it's being secreted, will form a gradient. And this gradient is what determines what kind of neurons the progenitor cells in the neural tube will differentiate into. And in addition to secreting sonic hedgehog, the floor plate also plays a role in making sure that the neurons have axons that are forming the right connections. So if there are defects in the floor plate, mutations in genes that affect the floor plate, this could lead to less sonic hedgehog production, which could affect the gradient, which can ultimately affect neural cell differentiation and potentially cause spinal malformations and birth defects. So the two genes that I decided to investigate, I decided to look at these because they're expressed very specifically in the floor plate and nowhere else in the neural tube, and they are present in both humans and zebrafish. And while foxA2 has been characterized in the spinal cord before, the role of spondin is largely unknown in the floor plate and it has not been previously investigated. So my research question is, what is the role of these two genes in the two main functions of the floor plate, which is the ability to express sonic hedgehog as well as the ability to guide axons? And so to do this, I used fish that had a not functional version of the gene of both spondin and foxA2, and these fish were bred, and the embryos that I collected, I used to run different staining techniques or staining experiments on them. So the first characterization method, the first staining technique, was in situ hybridization. And what this does is it labels mRNAs of interest, so genes that make an mRNA, and then we are trying to identify those mRNA and how much of the gene is being expressed within the fish. So here, all of the purple that you see are specific cells within this fish, this particular fish, that is expressing the sonic hedgehog A gene. And so in the spondin mutants, we didn't really see a difference. However, in the foxA2 mutants, we saw that they lost a lot of their sonic hedgehog expression, which implies that maybe there's also a difference in the amount of sonic hedgehog protein that they're making, which would ultimately affect the gradient. And the second characterization method I used was an antibody staining. And what we did is we put a fluorescent tag on the axons within these fish, from the developing neurons. And so for the spondin mutants, we found that they had fewer axons crossing the midline than wild-type fish, and we found a similar pattern for foxA2 mutants, which is important because axons crossing the midline are important for connecting the two sides of your body during development, and it looks like there's less of that happening in these mutant fish. So to conclude, it looks like foxA2 and spondin1b, these genes are both important for axon guidance. It looks like spondin1b plays less of a role in that sonic hedgehog gradient, while foxA2 plays a more prominent role. And for future directions, I would conduct those antibody staining tests again, but maybe with more neuron-specific antibodies, because the antibody that I used was more generally for a structure found in axons, but it could also have the odd staining for other cellular structures. And another future direction would be to analyze the protein expression now instead of just the mRNA expression. And so these findings will ultimately help to inform future therapies, future research on embryonic spinal cord like malformations and conditions. Lastly, I'd like to thank all members of the Huang Lab for their support and guidance, and I'd also like to thank the O'Brien Center for the BHSC for funding this project. And thank you for watching. If you have any questions, please do not hesitate to reach out.